All right, so this is the third video of your DNA foldable. This is the middle page. It says DNA at the top, and we are going to write what DNA stands for underneath. It stands for deoxyribonucleic acid. I'm going to take my little highlighter and I'm going to highlight the D, the N, and the A so that you know that DNA is short for deoxyribonucleic acid. Nucleic acid, which is an Na. So they match together with our biomolecules. Of course, the monomer of a nucleic acid is a nucleotide, which we just saw about. So here's our strand of DNA. And this, each part right here is one nucleotide. So in this section, we have 1, 2, 3, 4, 5, 6, 7, 8 nucleotides. 4 connected on this side, 4 connected on this side, and then they're connected in the middle. The sugar phosphate backbone, we previously have highlighted green, so we're going to go ahead and do this green. Okay, so here's our sugar phosphate backbone. This side part, remember, they're strongly connected together. They do not come apart very easily. And it's the phosphates and the sugars that attach together on this side, forming the long strand. Okay, then in the middle, we have our complementary nitrogenous bases, which we learned about. A goes with T and G goes with C. And right here we have our hydrogen bonds. These are our very weak bonds. They're easily torn. We're going to do those in purple. So between each of the bases here, we have these bonds that are kind of like Velcro. They're very easily broken. And when DNA needs to be replicated, or if they need to make a copy of a section of DNA to make a protein, they just unzip and they copy, and then they can go right back together, or they can split and become 2 separate strands of DNA. Okay, the very last thing that we're going to do is just color in our nitrogen bases based on the key that we made on page 1 right here. So anywhere you see an adenine or an A, we're going to color that in with blue. So I have an adenine over here, and adenine over here, and then its complementary base is a thymine, which is orange. So we're going to color in our thymines with orange. Then our cytosines are yellow, so I'm going to color in all of my cytosines. There's a cytosine there and one down there. And then the base that always matches with cytosine is guanine, which is a pink. So I'm going to color in, oops, missed one. My guanine's like this. Okay, so this is what your DNA strand looks like. This is a very, very, very small portion. It would be much, much longer. This part in the middle, this part is the actual code. Okay, the sequence of the bases is what is important, like the sequence of letters in a word is what gives the words their meaning. This is the part that's the code. This part is just holding it together. Okay, so once you have completed these 3 videos, and what you're going to do is you're going to cut on these dotted lines, and then you are going to tape together these 3 pages. Okay, you're going to tape together these 3 pages after you've trimmed them out, and then we're going to be taping them into our notebook. So once you have trimmed those pages out, you're going to get your notebook, and you'll see here I just taped them on the sides right here. So I have this fold out. I can fold it like this and fold it like this. This middle part is what's stuck on the page. And then I want you to label the front, my notes on the structure of DNA, and then a tiny little bit of RNA. It needs to be labeled so that when you look at it, you know what it is. Okay, so this is a big fold out. You're gonna tape these pages together like this. Then it should be able to fold up just like this in your notebook, and then label the front.